Hi everyone, my name is Lina Brilliantova and I'm going to present our work, a network-based approach for modeling gene regulatory relationships, joint work with Hannah Miller and Ivona Bezakova. Gene regulation is a process of control in which genes in a cell's DNA are expressed and produce their products, which in turn can themselves serve as regulators for other genes and so on. Gene regulation defines the properties and functions of every single living cell, providing us with the keys for understanding the mechanisms of disease and evolutionary processes. Gene regulatory relationships can be naturally captured by a gene regulatory network or a graph, discrete mathematical structure that has proven usefulness for various essential tasks. This is the example of E. coli transcriptional GRN with nodes representing genes and edges representing directed interactions between them. That can be activation if a gene's product increases the production of the target gene and repression otherwise. While the previous works analyzed the GRN topologies, the mode of interactions in GRN have not been formally analyzed yet, and this is the focus of our work. We propose the following probabilistic generative network models to explain the logic by which the gene regulatory interactions are formed. The first one is inspired by the Erdős-Renyi model. In this model, each edge is colored as an activator with probability PA and as a repressor otherwise. The model implies that regulatory interactions are formed randomly and that each gene will participate in the same fraction of activation regulations. Real-world GRNs, however, do not seem to have this universality and instead exhibit more complicated patterns. Our other models suggest that each gene itself has a latent regulatory type that affects its interactions in a system. In the source-consistent model, genes tend to activate or repress other genes. So the outgoing edges will be affected by the regulatory type of the gene. In the target-consistent model, genes tend to be activated or repressed. So therefore, the regulator type affects the incoming edges and the associated probabilities. For these two models, we also considered modifications called degree-consistent models to distinguish the effect of highly connected genes from poorly connected ones. In those models, the actual probabilities are the functions of the node's degrees, or the number of regulatory interactions that the genes have. It could also be the case that the regulation between a pair of genes is controlled by both gene types. That would, for example, be the case if we expected to see that activators tend to activate other activators and repress repressors. We model this type of behavior in our most complex model called binode consistent model, in which we have 4 different probabilities of having an activation edge, one per combination of activator and repressor. For this project, we have the following overarching research questions. Which of our proposed colored GRN models best describe the real-world regulatory relationships? And are those best models the same for organisms from different life domains? Each of our models generates a probability distribution over all possible colored GRNs, given an uncolored graph topology and model parameters. To address our first question, we need to solve the reverse problem. Given an input GRN from the distribution, find the model along with its parameters that has the highest likelihood of generating the network. Such model will be called the maximum likelihood model. As our input, we chose 2 publicly available datasets with transcriptional regulatory interactions of E. coli and mouse. For all models except Erdős-Renyi, we conducted a brute force search over possible parameter values. For ER, we found an analytical solution. The most computationally challenging model was binode consistent model, and we used Markov chain Monte Carlo sampling to estimate its parameters. We also reduced the number of combinations to test for that model by making the search space more coarse. For each model and dataset, we got the best parameter combinations, and from them got the maximum likelihood model with the corresponding parameters and the gene regulatory type assignment. And here is the results that we have. The best 2 models turn out to be the source-consistent and the binode-consistent models for both datasets, which is rather surprising since bacteria and eukaryotes have very different gene regulation mechanisms. On the closer look, we can also see that the fitted parameters of a BNC model basically turn it into SC because these 2 pairs are identical, meaning that a gene predominantly performs the same type of regulation to all of its target genes, regardless of their types, meaning that an activator tends to activate and a repressor tends to repress its targets. Interestingly, the equivalent of our observation was established for brain neural nets, where a neuron performs the same chemical action at all of its connections, which is also known as a Dale's law. What is also interesting about this table is that the fitted probability values are almost identical for E. coli and mouse, except for the fraction of activators. Here is how both datasets look like with the fitted latent regulator types. Each gene was assigned a probability of being an activator, and if that probability was greater than 1/2, then it was assigned an activator role, a blue triangle, and if it was less than 1/2, as a repressor, a red diamond. If you look closely into the graphs, you can see that there are also a bunch of undefined nodes for which their probability was exactly 1/2, and that those nodes are usually those that have very few connections in the system. So to sum up, we designed the first network models of gene regulatory mode and developed scalable algorithms for fitting their parameters and finding latent gene regulatory types. And we also found out that the source consistent model has the best explanatory performance for both datasets, and our maximum likelihood model can be used as a null model for simulating realistic gene regulatory networks, for example, in the creation of benchmark datasets. All the algorithms that we created for finding an ML model can be extended to also other novel colored GRN models. For the future work, we plan to use the gradient descent and replace the inefficient brute force search with it, run our experiments on other publicly available datasets, and also test the predictive performance of our maximum likelihood model to derive missing mode of regulation for partially known GRNs. Thank you very much for your attention, and if you have any questions, please reach out to us, and I'm looking forward to seeing you all at the conference.